MC26 of BSMT1, and I will be presenting a demonstration of DNA extraction of cheek cells here at home. Now, I will be presenting the materials used in this presentation.
Cool 70% isopropyl or ethyl alcohol. Food coloring. Dishwashing liquid. Salt. Cups and mixing equipment. Shot glass.
Okay, those were the materials used in the presentation. The first thing that we will do is dissolve half a teaspoon of salt in warm water. Open here. Now, pour some saline solution in the shot glass. Now, gargle the saline solution in the shot glass. Make sure to properly swirl inside the mouth the saline solution for about 1 minute. Remember to spit the gargled saline solution in a separate glass. Now, add 2 drops of dishwashing liquid in the spitted saline solution. Gently stir the solution and remember to not make any bubbles. Hindi mo na, may green pa. Anak na. Next is... Now, in a separate glass, add half a cup of isopropyl alcohol. So these are... In the isopropyl alcohol, add a pinch of food coloring. Church na lang sa buhay ko. Now in the spitted saline solution, gently pour the alcohol mixture.
What is the purpose of adding dishwashing liquid in the solution? The purpose of adding dishwashing liquid in the solution is to disrupt the cell membranes by pulling apart the membranes, releasing the DNA inside. Dishwashing liquid is a detergent that is capable of breaking down fats. On the other hand, chick cells are made up of cell and nuclear membranes made up of phospholipids. Thus, the soap is capable of breaking that down, causing cell lysis of the cell. What is the purpose of adding isopropyl or ethyl alcohol to the solution? The purpose of isopropyl or ethyl alcohol to the solution is that it clumps together the DNA and allows for the formation of a white precipitate. It's important to use cold alcohol because it allows a larger amount of DNA to be extracted. If the alcohol is too warm, it may cause the DNA to denature or break down. Describe the appearance of the DNA that you extracted. Why is that the DNA appears to be loose strands and not compact? The appearance of my DNA looks whitish in color and is very stringy. It appears to be loose or stringy because of the addition of alcohol. DNA does not dissolve well in cold and concentrated alcohol. So when the DNA molecules precipitate in the alcohol, a number of DNA tangle and clump together, forming strands on top of the solution. This kitchen methodology has some of its limitations. Explain which steps from the standard DNA extraction procedure you skipped while performing the kitchen methodology of DNA extraction. The step that we skip over while performing the kitchen method is DNA purification. That primarily makes sure that the DNA samples are not contaminated. Enumerate the factors which you think might affect the yield of the DNA extracted. Factors that might affect the yield of DNA extracted are too much salt or not enough salt in the saline solution, improperly mixed dishwashing liquid in gargled saline solution resulting in incomplete lysis of the cell, and alcohol was poured at a fast rate. After DNA extraction, you were tasked to store the DNA so you can use it in other techniques such as PCR and electrophoresis. Explain how will you store the DNA. To properly store the extracted DNA sample, I will first collect the DNA using a toothpick and then place it in another sealable container that contains alcohol. Then it will be stored in a refrigerator.